Good So, morning, sir.
Good morning.
Yes, sir. Good morning, sir. So, we'll be starting it shortly, sir.
Pardon?
We'll be starting the session shortly, sir.
We may start.
We have a much to join, so maybe we can start that, sir. We have Dr. Um, we have Chaudhary ma'am. We have Momita Chaudhary ma'am.
Uh,
We can wait for another 5 minutes. And our principal has also not joined. Narendra sir, Sanjay sir, when he will be joining, sir?
Hello, wait a minute.
Good morning, Sanjay sir.
Hello, sir.
Good
Good
morning, sir.
morning.
Good morning. Good morning.
Uh, we are happy to collaborate.
Yeah, yeah. Thank you, sir. Keystone.
Dr. Govindaraj, sir. He is a principal and professor from Wells Pharmacy College. Welcome, sir.
Thank you, madam.
Good morning.
Yes, good morning, sir.
Good morning.
Good morning.
Thank you accepting our invitation.
Yes, yes. Thank you, sir. Thank you, sir.
Uh, uh,
So today we have Pratik with us. So Pratik will be the host for today. Pratik will be starting with another 5 minutes. Just let me ensure that Chakravarti ma'am will join you.
Shoaib, sir.
yes, sir.
Ah,
Yes.
yes. You can start now.
Yes, yes. Okay, sir. Hello, dear sir. Proceed with session.
So Mamata ma'am has not received the session. So, so let me call her and we can start. Sir. You can start the session. Pratik, you can start the session.
Yes, I'm here only. Okay. So everyone is here, I guess?
Yes, ma'am.
Okay, let's give a moment. Well, good morning, everyone. And uh, welcome to this bold new age of cure where science unlocks what's what not secure. From targeted cells to precision arts, you mend the body and uplift the heart. A revolution brewing in every pill to answer mankind's most enduring will. I, Pratik, welcoming you here, and it is our pleasure to welcome all our participants, guests, and organizing team for a two-day international conference on pharmaceutical industry revolution, a focus on innovation and research, which is organized by Academy Innova World in collaboration with H.R. Patel Institute of Pharmaceutical Education and Research, Shirpur and ISTEM. Academy Innova World, it is a hub for research-driven innovation, technical excellence, and intellectual property creation. Founded in 2017 as a YouTube channel for aerospace enthusiasts, it now holds an MSME registered STEM research center recognized across India. Unlike conventional academics, our academy operates a dedicated R&D cell collaborating with researchers, scientists, and industry mentors and other candidates, a rare offering in the education sector. Recently, Academy Innova World has been declared as India's first private research and innovation hub, which was featured by Republic News and Gujarat Journal. Academy Innova World is happy to collaborate with a renowned institution like HRPIPER to accelerate innovation, foster crucial collaborations, and translate groundbreaking research into real-world therapies that improve global health outcomes. The pharmaceutical industry is revolutionizing through biotech, AI, and personalized medicine, aided by regulatory reforms. The International Conference on Pharmaceutical Industry Revolution united scientists, researchers, policymakers, and healthcare experts to explore innovation in drug discovery, manufacturing, and patient care, shaping a transformative future for medicine. Now, I extend my warmest welcome to our esteemed dignitaries, Dr. Aishwarya Dhara, founder of Academy Innova World, whose visionary leadership has been instrumental in driving innovation and excellence in research and education. E.R. Pulak Kumar Dhara, Chief Technology Officer and Organizing Chair, former Assistant Scientist also at the Department of Atomic Energy, whose expertise and dedication continue to guide our technological advancements. Now, Dr. Sanjay Kumar B. Barry, Principal, HR Piper, Event Coordinator, Mr. Narendra B. Patil, and Mr. Shoaib M. Manyar, HR Piper. I welcome our keynote speakers of the day, Dr. G. Sarvanan, Professor and Principal, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Science, Wells Institute of Science, Technology, and Advanced Studies, Tamil Nadu. Now, moving forward to Dr. Moumita Chaudhary, Assistant Professor, Guru Nanak Institute of Pharmaceutical Science and Technology, Kolkata. Once again, I welcome one and all present here. Now, it's time to call upon Dr. Sanjay B. Barry, Principal HR Piper, to deliver an inaugural address. So, please, sir.
Thank you, Mr. Pratik Ranjan, for welcoming all on this occasion. Am I audible, sir?
Yes, sir. Yes, sir.
Thank you. Thank you. So, good morning. On the behalf of HR Piper and Academy Innova World Kolkata, jointly organized on this online workshop on the International Conference on the Pharmaceutical Industry Revolution and focus on innovation and research. So it's my pleasure to brief about the HR Piper, which was being established in 2004 under the Shirpur Education Society. We are catering Diploma Pharmacy, Bachelor of Pharmacy, Master of Pharmacy with the 4 branches: Pharmaceutics, Quality Assurance, Pharmaceutical Chemistry, and Regulatory Affairs, as well as the PhD approved center by the Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon. Our institution recently accredited by NAC with A grade. We have been NBA accredited twice. Our institution is growing, and our faculty and members has been received the different research grant, more than the 5 crores grant from the various agencies. And we are moving towards the academic excellence with the current updates. And the objective behind this keeping the online webinar is to update and upgrade to the student community or a scientific fraternity. And that's why I am thankful to the Academy Innova World, all the dignitaries, those who are present here, for the sponsoring such a wonderful webinar for the benefit of the scientist, student community, research community, fraternity. And I also welcome today's expertise, Dr. G. Sarvanan, as well as the Dr. Moumita Chaudhary, for their valuable time. And definitely this webinar will be the fruitful for the addition in this particular innovation and research. And really it will be useful for everybody. So I am very thankful to the Innova Academy for sponsoring such a programs, and we expect same cooperation in future also. Thank you very much. Thank you, sir.
Thank you very much, sir. Now moving forward towards it, it's my pleasure to introduce our keynote speaker of the day, Dr. G. Sarvanan, Professor and Principal, Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Wells Institute of Science, Technology, and Advanced Studies, Tamil Nadu. Dr. Sarvanan sir specializes in novel drug discovery with an H-index of 25 and over 1,953 citations. His impactful research significantly advances therapeutic science and demonstrates his esteemed authority in the field. Today, he will share a few insights on the QSAR revolution, designing better drugs with productive sciences. Over to you, sir, please.
Yes. Thank you, sir. I hope I am audible.
Yes, sir, you are.
Yes.
You
Very
can
good
take
morning
over.
to one and all. So, on behalf of that Vale Pharmacy College and Vistas, first I would like to thank that HR Patel Institute of Pharmaceutical Education and Research and Academy in our world for giving this great opportunities to interact with that young pharma peoples. And particularly, I want to thank that convener, Dr. Sanjay Kumar B. Bari, Professor and Principal, HR Piper, and Engineer Plok Kumar Dara, Chief Technology Officer, Academy Innovaval, and the coordinator, Mr. Narendra B. Patel, and Mr. Soyab S. Manyar, HR Patel, Institute of Pharmaceutical Education Research, and Dr. Aishwarya Dara, Academy Innovaval, for giving these great opportunities. And without wasting much of time, that I will share my screen and I will start my presentations. I hope my screen is visible.
Yes, sir.
Yes, sir.
Thank you. So today, that the topic which I was chosen, which was aligned with the title of our conference, that quantitative structure activity relationship, and what this QSR is a revolution in the designing of a drug molecules, because it is a nowadays that it made a revolution in the predictive science. So hence I chosen the title as the QSR revolutions, designing a better drugs with predictive science. So in my presentations that I'm going to cover what is quantitative structure activity relationship, and if you want to do on a work or using an QSR, on what way, what step by step we need to go into that work in that QSR, that workflow I'm going to discuss, and how it will be helpful in the designing of a drug molecule in the drug discovery. And ultimately, when any new technologies or any technologies we are using, ultimately the technology will have some challenges and limitations, and what are all those challenges and limitations which are associated with this quantitative structure activity relationship. And if time permit, I will go ahead with case studies. Okay, first in the QSR. So we must know what is this QSR, and whether that QSR is a new thing, or what are all the important milestones in the history of a QSR, and what are the basic principles which involved in the QSR, and what basis this particular technologies are working, and how it will play a role in the designing of a drug molecule. That only initially we are going to discuss. First, what is QSAR? The name itself it is that QSAR. That is, Q indicates quantitative, S indicates structure, A indicates activity and a relationship. So there is some relationship between the structure and a biological activity. Okay, and if we are able to quantify that relationship between that chemical structure and a biological activity, that is called as QSAR. In a scientific way, if you want to tell, means that we are using a mathematical modeling. Okay, that mathematical modeling, what it is relating, here I told relationship between a structure and activity. So we are using a mathematical model. The mathematical model can able to relate that activity with on a structure, or that activity with on a physicochemical activity, and how it will relate means by means of using a computational tools. So by using a computational tools, if a mathematical modeling is able to relate that chemical structure and a biological activity, that is called QSAR. So in a simple manner, if you want to tell, means if any molecule is there, how that molecule will biologically behave, or what that molecule will do when it is enter into our biological system. So that after taking a medications only we came to know, but by using this particular technology we can able to predict. So if we are taking this particular molecule inside the biological system, it may produce such type of activity, like that it will give on a predictions. In a simple form, in a very simple form, if I want to tell that what is QSR means, whenever if you want to have on a food, or if you want to taste the food, initially we will ask what are all the things are ordered. Okay, for example, if it is on any sweets are there, okay, so initially if we came to know, okay, what the things was ordered, the sugar was ordered, the ghee was ordered. Okay, so in case based on that, the ingredients what we are adding, the taste may be differ. Okay, so to that sweet, when we are adding on a cashew nut, automatically what will happen, the taste may increase. Okay, so like that, this QSR is on a some mathematical recipe we can call. Okay, so that mathematical recipe what it is doing means it will link the chemical structure and how that chemical structure will changes or performs the biologically. Okay, so that is the way how we can simply understand about QSR. If you are looking at a history, that the concept was not started yesterday or day before yesterday. It was started in a long back in the year 1860. The concept of relating a chemical structure with a biological activity was initiated by a scientist called Crum Brown, who's the scientist who related that chemical structure with a biological activity. And what he proposed is that physiological action, any molecules, any drugs if we are taken, what that biological activity it was producing, it is depends upon their chemical composition and structure. After a long year, 100 years, the 1964, we can call that father of QSR, who is none other than Hans. His full name is Corwin Hans. Okay, he introduced that equation, which is a well-known equation. It is an Hans equation. What this Hans equation is doing means it is mathematically link our the biological activity with a physico-chemical properties. That physico-chemical properties like electronic properties, steric factors, hydrophobicity, like these physico-chemical properties of a drug molecule was mathematically related with a biological activity. And from that 1970 to 1990, and there are databases are developed, chemical databases, and computational powers are coming into that usage. Because of that, that in that periods, this particular technology was used to optimize the drug candidates. If so many compounds are there, if 100 or thousands of compounds are there, from that which one we can optimize. For that purpose, they got earlier used in the year 1917 to 1990s. And from 2000 onwards, at present, what things was changed means nowadays if we are going everywhere, the word what we are hearing is on a AI. There is artificial intelligence. Now along with that, the another one word what we are hearing is on a ML, machine learning. Okay, so why can't we integrate these artificial intelligence and machine learning with a QSR? So now they are integrating that particular thing, and they are extending the application of a QSR into the toxicology as well as personalized medicine, environmental science. In all these cases, that the QSR is expanded, and even regulatory bodies like US FDA is started considering about QSR data if we are performed well, okay, for toxicological evaluations. Okay, in a simple word, if you want to tell me, it's QSR, which will bridge us the chemistry with biology. Okay, chemistry is there and biology is there. The QSR is on a bridge. Okay, and on what ways means with predictive precisions. Precisely, it is predicting if that chemical nature of the particular compound is like that, the biological activity will be like this. It is predicting. Okay, sir. So, as we know from this introduction that a chemical structure and a biological activity, there is some relationship. Okay, if it is a relationship, how? What is the basics behind these biological activity and chemical properties, biological that property of a particular compound? Okay, so if you are looking that all you people may aware of that one dimensional, two dimensional and three dimensional, four dimensional structures. Okay, and if we are taken on any molecule in the three dimensional way, if you are looking the how that atom was arranged, how that functional groups are arranged in. Okay, because of that arrangement of an atoms, arrangement of an functional groups, their electronic properties and their shapes will change because of that changes in the structural features. Okay, so for example, you think that there is a carbon and there is a one hydrogen was above and there is a three methyl groups are in the down below the plane. Okay, in another cases you can think that there is a carbon and one methyl groups are above the plane, that two other methyl groups are below the plane and one more hydrogens are present below the plane. Then automatically what will happen, the first compound can interact with that biological systems in one manner and the second compound can interact with the biological properties of another column, another way. Okay, the same thing will be applicable to one drug molecules also, because the drugs can go and interact with that biological targets. Biological targets means here it may be a receptors, enzymes, etc. Okay, because the biological targets will have some specific structures. Okay, so if our compounds have able to interact with that particular one, if it have a similar that can able to have such particular structures only that can able to go and interact with that one. So because of that, the structure will relate to the biological activity. Okay, so if targets are there and we have drug molecules. Okay, so we planned we designed in a molecule in such a way that can able to go and interact with the biological targets properly. Okay, when it is interacting with the biological target, what may happen? When it is interacting with the biological target, the activity may increase or it may decrease, or the totally the compound can be inactive. And the same thing will happen to one toxicities also. The toxicity may decrease or it may increase. Okay, it is depends upon how it was interacting with a biological target. So the basic concept is what? Molecules, when it is looking in on a 3D arrangements, they have a different properties. They have a different shapes. Because of the different electronic properties and a shape, that molecules can differently interact with biological targets. A simple example we can take it. So take an example as a benzene. Everybody know that there is a 6 carbon and 6 hydrogens are there in a benzene. And we think that there is a one hydrogen atom we are replacing with a chlorine in the benzene. When we are replacing that one hydrogen with a chlorine, what may happen? It may happen to a potency, that biological activity, okay, that can increase or decrease or can be inactive. Similar thing may happen to one toxicities also, or the total molecule may become inactive. So in QSR modeling, what they are doing? So they are converting the structure into a numerical descriptors. Keep remember. If it is QSR, the first thing you must remember about is descriptors. Okay. How? What is that example for a numerical descriptor? So each molecule have its own molecular weight. They have its log P value and may have some number of hydrogen bonds donors or some number of hydrogen bond acceptors. All these things molecular weight. So for example 115, it is what? A numerical number. Log P value, it may be 8 or 9. Again it is a numerical value. Hydrogen bond donors 3, 5, 1, 0. Again it is a numerical. So what we are doing in the QSR modeling means keeping the structures and we are converting that structure into these numerical descriptors. Once we are converting that a structure into a numerical descriptors, then that numerical descriptors only we are going to use it to predict its biological activity. Okay, so how we can relate with on a normal day-to-day life? Okay, so before go build any building, what we will do? We will make it as a blueprint. Okay, similarly, before producing a biological activity, the structure of the molecules, okay, which will dictate how that molecule will behave biological systems. Okay, so chemical structure is like a blueprint. Based on that only it can go on produce the biological activity. In blueprint, if you are changing, then automatically what will happen? The total building will change. Similarly, if you are doing some changes in a chemical structure, then automatically the changes will take place in biological activities also. That is the basic principle you should understand about a quantitative structure activity relationships. So if you are looking in the drug design, this QSR, what it will do? It will reduces the cost and the time. It will guide a medicinal chemist in order to select some promising compound. So for example, when I was doing an UG and an PG in the year 2002 and 2005, that time these techniques are there, but it was not that much used. So usually what we will do means in the laboratory, day to night, sorry, day to night we will work and we will synthesize some 10 compounds or 15 compounds in a series, and all those 15 compounds what we will do means we will test its biological activity. And if you are testing all those things, now maybe one, sometimes none of the compounds may be active, but you look at how much time we spend there to synthesize that particular molecule, and after that it's for a biological activity. But this QSCR is reducing those things, and it will help that medicinal chemist in order to synthesize only that particular active compounds. Before going to the wet lab for the synthesis, we can able to out of that 15 or out of that 100 which compound is more active. Okay, no need to synthesize that all those 100 compounds, omitting those 99 compounds which are not fit into that cases, and only one compound which have shown a good activity, that compound only we are synthesizing in the laboratory. And because of that, what's are happened? Because of this, I don't need to spend a lot of time in a laboratory. Okay, I can able to concentrate on my experimental work, and because of that I can able to find out on a many lead molecules, and hence that efficiency in drug design and discovery will increase. Okay, that's why we can tell that QSR will play a major central role in the modern drug discovery process, because we can call this one as a predictive engine. It will predict if you are giving some drug molecules, it will tell it on what way it will behave in the biological systems. Okay, and hence it is one of the major tool that medicinal chemists are using before going into the lab experiments. So this QSR will play the role in virtual screening of your compounds. From that we can able to identify a lead compounds. Once the lead compounds are optimized, then we can able to test its toxicity, ADMET properties we can able to predict, and from that we can able to go into the decision making, by which many data are collected. From that we can go for a data driven decision making. In all these cases that a QSR will play a major role. Now coming to that workflow, how that work is going and what sequence we need to do work when we want to plan for a quantitative structure activity relationships. Okay, first thing, as I already told, it is on some mathematical modeling. Okay, if you want to create that mathematical modeling, we must have some data. Already published data are needed. So from where we need to collect that data. So that is the first and foremost important step in a QSCR, data collection. Okay, after collecting the data, we are converting that structures into a molecular descriptors And by using a feature engineering techniques, we are making the data that we are sharpening that particular data. After that one, we are developing some model for that a particular data sets by using a machine learning models and using several statistical tools. Then once it was developed, we need to validate that a particular model which was developed, and we are analyzing its performance matrix. Okay, we will discuss one by one elaborately. Data collections. Okay. So as already I told, it is the very critical step, because these are the input to the particular study. When you are not giving input correctly, you can't expect that output correct one. What you are giving to the machine or on a system, for that only the machine or system will reply to you. Okay, so we should take care what commands and what inputs we are giving into the systems. So this is the most critical phase. Okay, so here what we are doing, we are collecting the chemical structure and biological activity data. And biological activity data, what we will collect it usually from the published data, published papers, we will take an IC50 value, inhibitory concentrations, and effective concentrations, okay, Ki value like that, to on a specific targets. So for example, some HMG-CoA inhibitors or some receptor beta blockers, okay, alpha-2 receptors, like that, for some specific biological targets, we are collecting the data which are already published and which are already available. Okay, so for that one, what are all the things we need to consider? First thing, we need to choose your biological targets. Even though for a same disease, there will be many targets. Okay, in that, which target you are choosing, that is the more important. And whenever you are choosing a target, first thing what you should keep in your mind before going to on a QSR is whether there is a ample amount or sufficient amount of your data are there to that particular targets or not. That is the thing here I have given you a tip. Choose the target with an ample amount of published activity data. You can choose on any receptor. So for example, acetylcholinesterase, epidermal growth factor receptor, okay, or SARS-CoV-2 MPRO receptors. But the target you chosen doesn't have any data, published data, then what happened? We can't be able to develop a particular model. So before collecting the data, choosing the targets, we must aware of that whether a sufficient data are available for that a particular data, particular targets or not, we should confirm. Second case, sources. Okay, there are so many sources are there. You must aware of that whether that a particular source is reliable or not. Okay, and again whether it is freely available or not. Okay, all those things we should consider. So these are all the some of the major sources what we can use it for our QSR studies. ChemBL, PubChem Biosay, Drug Bank, Zinc 15, Royal Chemical Society Protein Data Bank, all these things. And I was given which data it will be provided. So for example, if you are looking at a ChemBL, it will give you a data of a curated bioactivity with a smileys it will give. And that is the source which will best fit for a QSR. So like that, the other sources and what data it was providing and what we can get it. So that was given in a table form. Once we get identified on a targets, then where we are going to collect the data. Now it is our time to download the data. So for example, here I get taken ChemBL source. Okay, we need to go to that particular website, and there I am searching on a target. Here my target is on acetylcholinesterase, and that have some particular ID number in that one. and from that I'm navigating to on a bioactivity steps. In the bioactivity steps, I'm selecting and a filter what are all the things I needed in the bioactivities. Okay, I need an IC50 values, nanomoles, and which assay type, binding assays, like that. Once after selecting all these things, the most important thing is we need to download the data, usually as a CSV file. That is nothing but CSV file is the values are separated by a comma. That's all. Okay, so what these data CSV file will contain means it will contain that chemical ID of that particular compound. For that compound smileys, there are IC50 values and a target name, what is that one, and which assay type it was performed that particular activities. Okay, so now I selected on a target, I downloaded on a data, and now I need to prepare my data sets. Okay, so what we need to do, we need to prepare tables like that. So compound in one column, smileys in another column, and like that IC50 value and whatever you collected from the data sets, that we are keeping in a separate columns. And if it is an IC50 value, in order to for a convenience purpose, okay, we are taking that logarithm to an IC50 values, and we are converting that IC50 value into an PIC50 value. Okay, why we are converting this on IC50 value into a PIC50 value means that is it is easier to compare its effectiveness when it is on a PIC50 value than normal IC50 value. Next step, we need to generate a descriptors. Already I told descriptors are the numerical values. I told what is that? Hydrogen bond acceptor, how many? Molecular weight, what is the molecular weight of a particular compounds, like that. We are converting that descriptors into numerical values, and we are using that smileys, what we collected from our sources to calculate the molecular descriptors. And for that one also there are so many tools are available like RDKit, PADL descriptors, ModRed, CDK descriptor calculator. All these things can be used to generate a descriptor. And now the data sets will be like that. Okay, first the compound will come, then after that with comma, that is the thing I told, CSV file, then smileys, IC50 and in value in a nanometer, PIC50 value, molecular weight, like that, these are all the things one value. This is the compound name. Okay, this one is on a smileys, and this one is an IC50 value, this is a PIC50 value, like that. Okay, the sets will be arranged. Then if you want on a good QSR data, what criteria it should match? Okay, it should have compounds, okay, more than that, at least 30 compounds. And so for example 10 compounds data I got collected from one activity. So for example, if it is an analgesic activity, okay, in the tail flick method I get taken 10 compounds. In another method, okay, polyking method, second cases, if I'm taking, then it will not be on a good QSR data. Okay, because that is the one method, this is the another method. So we need to select the same assay type. Okay, even though the target is same, which assay they get performed, because that assay changes also will make some influence in the data. Because of that, we need to select from a same assay type and the same target we need to take. And it is the responsibility to clean our smileys before giving an input. And always it is better to use PIC50 value instead of inhibitory concentration 50. So these are the molecular descriptors, how it will be calculated. That molecular descriptor is a numerical value which will represent a specific property of a particular molecules. Okay, and these molecular descriptors are the input for a QSR. Keep in mind, these are the features which we are going to use in a QACR model, that it is nothing but it is an input. So these are all the five different types of descriptors which are available, and the example and what it will give. That description I was given in the table. So for example, 3D descriptors, for that one we need a 3D structure. From that 3D structures, what we can get it means we can able to get a surface area of that particular molecule and volume of that particular compounds we can able to get it. Like that, if we are going to on a quantum descriptors, we can able to calculate an high occupied molecular orbitals and low unoccupied molecular orbital energies, and these things can be used for an density functionality theory, that generally we will call it as a DFT. And these are all the tools, what language they are using, how it will be interfaced, and on what ways with some specific notes I get given for your easy understanding. Okay, so if it is on a Padal descriptors, and it may be some approximately 1800 descriptors will be available. Like that data barrier, we can use it for an quick and visual analysis, we can use it. And for chemistry development kit, CDK, that we can be interfaced with an API and GUI. And okay, now we selected on a target, we collected, downloaded, and we set on a data sets. After that, what is these feature engineering steps? Okay, we are making the data is proper or not. So in order to make the data as a proper data, we are undergoing these feature engineering steps. Okay, so first we need to clean the data. Okay, we need to check whether any values are missed or any zero variants are there, and whether all that IC50 values are converted into on a PCA, that logarithmic IC50 value or not. Like that, we are cleaning the data. Once it was cleaned, then we are normalizing the data, because many machine learning models are very, very, very sensitive to the scales. Okay, so hence we are normalizing that data is what we collected. Then from that normalized data, we are choosing which relevant features we needed to study. Okay, so for that purpose we are using three different methods: filter method, wrapper methods, and embedded methods. Okay, and a filter method is based on the correlation or on a variance, and wrapper method will use a model performance, and embedded methods will build into on a models. Okay, so we are cleaning. After cleaning that, we are making that one to a normalized. After normalization, we are selecting the sum of the features which are available in that particular structures. Then we are reducing the dimensionality. Okay, that is the principal component analysis. You look like that it was there, but if it is we are using as such, it will be difficult. That's why what we are doing, from that which area we can take one part of that area, we are taking in all the compounds. A two compound, that's a two, like that for all compounds. Again in that in one direction, if you are taking a smaller one, so that's why you look dimension how it was reduced from this compound to here. Okay, we need to follow the uniform things for all compounds when it is going into a dimensionality. So finally we are getting the data like this. It is the molecular weight, log p value, this many hydrogen bond acceptor, this many hydrogen bond donors. Okay, these are the IC50 value. Like that, the final data set will be available. Then we are making into a problem setup. Okay, there is on two types when it is coming into on a problem setup. One is on a regression type, the another one is on a classification type. Okay, so for here what we are giving means we are giving some input features. How we are giving that input feature means by means of molecular descriptors or on a fingerprint, that is the input, And after that, we are making into an PIC 50 value. In that, based on that, we are making that one to two different class of groups as an active and an inactive class. This is the way how we can select that model. Different models are there, and that model will come under which type of things, whether it is a regression type or a classification type, what we discussed here. Okay, so these are all the various models: random forest model, logistic regression, linear regressions, like that. Once that model we selected, then what we are doing, we are training that particular model. After training, we are evaluating. Okay, so we are gathering the information. After that, we are selecting the training model. Once it was a training was completed, we are validating the training. Okay, after validation, we are testing that particular model with another data sets which are not given during a training. By using that one, we can able to test our model. So validation, and there are 4 different types of validation here. It may be an internal validation, or it may be a cross validation, or it may be the external validation, or it may be a Y randomization type of validation. And each type of a validation will have some specific purpose. So for example, if you want to, when you want to test whether that model learns real patterns, or the model having any random noise, in that case you need to select a Y randomization type of validations. Okay, and for regression QACR models, this is the metric what we are using and how it will be helpful for interpretations. Okay, for classification models, and we are making labeling as an active or inactive labels here. Okay, in case of Y randomization, what we are doing means we are randomly shuffling only the Y values, not disturbing X values. Okay, and after shuffling the Y values, so for example, one data set I get given, I tested, then the same data set I am taking and I am shuffling only the values of Y, not on the X's. Then again I am retraining the model. Okay, and we are checking whether that accuracy is good or not. Okay, and whether if we have if we got a poor R square value, so for example 0.0092 like that, then that indicates it is a good model. If you are that regression coefficient is more, then that indicates it is not on a good model. And where that a particular model is going to be used, and that also there is 2 methods are there. One is on a leverage methods, the another one is on a distance based one. Okay, so till now we discussed about what is QACR and how we need to do a workflow in that QACR. Now we are going to elaborately discuss about what way this QACR is applicable in the drug design. There is 3 major areas where the QACR will play a major role. One is in optimizing a hit to lead compounds. The second case, if you want to predict the toxicity, and if you want to know about ADMET profiling, that also the QACR will play a role. And if you want to resist that drug resistance, and if you want to anticipate that side effect, for that purpose also that QACR will play a role. And elaborately we will discuss with optimization of a hit to lead. Okay, this is the one of the critical phase in a drug design and discovery. Okay, so once we have a hit compound, okay, and that from that hit compound, by means of a high throughput screening, that will optimize that hit compound into a more potent as well as a selective compound, and that compound only we are calling as a lead compounds. Okay, so from a hit compound only, after optimization we are getting a lead compounds. When we got a lead compound from that hit, then by using a QACR, we are predicting and we are improving the biological activities. Why we are doing here? Because it may have lead compounds have a desired biological activity, but it is not up to the mark. We need to increase its biological activity. In the meantime, we should consider about toxicities also. We need to decrease the toxicity. Okay, for that one we need to perform before going to the wet lab synthesis. And this is the steps we need to follow in the optimization of a hit to lead compound. First, we need to identify. After identifications, we are modifying structure. Why we are modifying structure means in order to increase the biological activity and in order to remove or expel the unwanted side effects. After modifications, we are predicting its activity. Once it was showing on a good activity, then we are optimizing that particular one. Then based on that prioritizations, we may work on that particular cases. And these are the common QSR technique what they will use it for a hit to lead optimization. We will use your 2-dimensional and 3-dimensional QSR. We will use your machine learning models for Forest SVM neural network for better prediction accuracy and descriptors. It includes the physicochemical and quantum chemical features. A simple example, if you want to do means in anti-cancer activity development, there is some compound which shows some moderate inhibition against epidermal growth factor receptors. Okay, how we need to approach that particular compound? For this EGFR target, we are collecting a data. We are making a data sets. After that one, we are calculating the descriptors. Once it was calculated, then we are training the. Okay, then we are using that model to add some structural features in order to improve its biological activity. Okay, because of that changes in the structures, that outcome may increase with increased potency and less side effects we will get it. Okay, advantages if you are looking, it will reduce the compound which are synthesized, it will save the time, and it will identify the trend of the structure activity relationship very quickly, and it will predict not only the activities, it will also predict safely in the parallel manner. That's why we can call the QSR as a powerful tool. Okay, we can use for select, design, and optimize the compounds efficiently by means of using a computational methods. Okay, and it is not only used for a prediction of optimizing the hit to lead compounds. Nowadays we are using that for predicting a toxicities also, and also we are using for an ADMET profiling also. We are started using this QSR. Again, this is the way how we can able to use it for toxicity predictions. For the target is an HERG. We are making a data sets, then we are making a model, then after that we are making predicting the risk with an HERG blockers. Then if they are high risk that particular compounds are showing, then it is that we can able to discard that particular compounds in the early stages itself, that it will save so much of time as well as money in the drug discovery process. So like hit to lead optimization here also, in the toxicity prediction also, it saves the time and reduces the testing, and it will identify the dangerous compounds, and we can easily integrate that one. So this is the goals, and in each stages how that QSR we can use, that was given in that particular table. And coming to that, anticipating drug resistance and side effects, as all you people may aware of that drug resistance. Okay, there are that is the one of the major problem what we are facing in the that antimicrobial therapies, that antibiotic therapies. So here resistance, what it is meaning means that a target organism is mutating themselves, or what that organism is doing means they are adapting some situations to escape from the action of a particular drug molecule. Okay, the reasons may be these things, because of the mutations or alteration in the metabolic pathway, or formation of a biofilm, or overexpression. Because of that, the drug resistance is happening. And like that, the drug may produce some off-target effect also. Okay, so these things can be predicted by means of using a QSR. So these are all the tools we can use it for anticipating the drug resistance. Swiss target prediction, AdMetLab, Moldesto, Pass Online, all these things we can able to use for this side effect predictions. Okay. And just another 2 minutes, I will complete my presentations. A lot of time was completed. Okay, that anyway, as I already told, any techniques, definitely new techniques are coming. Always there will be some challenges as well as there will be some limitations. And here, in case of QSR, looking that QSR is fully depends upon the data only. Okay, so the quality of the data and the availability of the data, there only we will face other things. And whatever the data which was there, if it is overfitted, then there will be a problem we will face. And acceptance by regulatory bodies, and whether that a particular model is reproducible or not. Okay, so this quality and availability of a data is on a very critical one. If the data are less available, then automatically what happen? The model what we are producing will not be an accurate one. And if your input is not correct, automatically we can't able to expect that output also correctly. Okay, so here number of compounds what data we are collecting, and quality of the data, whether it is a reliable sources or not, whether that particular data are reproducible or not. Okay, and data diversity. And that is the thing I told. If you are taking that unit, if one it is in micromole, the another one is in nanomole, we need to convert again both the things into any one unit. That way only we need to do it. So that data quality will affect accuracy, consistency, cleanliness, balanced class, and all these cases. These are all the factors which can affect that quality of an data. And usually what issues we will face in the data means we will do on a duplicate entries. There will be some false positives, and due to an experimental errors, the data may be wrong one, or some data cases we may missing values will be present. Okay, in order to improve the data, we need to use a curated databases, and we need to pre-process that data. We need to standardize the structures, and we need to always use a reliable assay sources only. We need to use it. So these are all the reliable public databases that already I was explained, Campbell, all those things. And go ahead with that overfitting. Okay, that means whatever the model you are selected, prepared is fitting into the one model perfectly, but in the another model it is failing. One data set when you are selecting, it will give on a 99% accuracy. But in case if it is giving only 60% for a new compound, then it may not be good. Okay, so that is the another problem what we can face in an QSR. This is the way how we can prevent that overfitting. Okay, by means of cross validation technique, external validation, by means of selecting a simple model, regularizing that particular model. By that way we can able to prevent that overfitting. Even if QSR is slightly less accurate, but if you can able to interpret it, that is good than a well validated model. Okay, compared to on a black box model, if your model is less accurate slightly, I'm telling it is not more, slightly. But if you are able to interpret, and if your model is well validated, that is better than the black box model that cannot be generalized. Okay, and the regulatory agencies are nowadays start accepting an QSR model. Okay, so hence if you that regulatory body has to accept it has to satisfy some of the conditions which was given by an regulatory models. Okay, so by that way if you are preparing your QSR data sets, that regulatory bodies can accept it. Okay, researcher and regulators are trusting that QSR model when if it should be predictive, transparent, accessible, and repeatable. Then automatically the regulatory bodies will accept. These are all the QSR tool which are approved already by the regulatory models. Okay, and with these things that I will come into my conclusions. So case studies I am not going. Okay, and if you have any questions, it was welcome. 5 minutes. Yes, thank you, madam. Once again, thank you all for giving these opportunities.
Thank you, sir.
Namaskar for such an informative session, and it was pleasure that you came here and enlightened us with the vast knowledge of QSAR, which is still in progress. And now, moving forward to this session, we have another keynote speaker, Dr. Momita Chaudhary, who is an assistant professor and a researcher at Guru Nanak Institute of Pharmaceutical Science and Technology, specializing in drug delivery systems with an H index of 8 and 296 citations. Her work demonstrates a growing impact in advancing pharmaceutical formulations and therapeutic efficacy. I request ma'am to share a few insights on this domain. Ma'am, please.
Thank you, sir. Am I audible?
Yes,
Yes,
audible.
ma'am, you are.
Good morning, everyone, distinguished delegates, esteemed colleagues, and participants of this conference. It is my privilege to be present at this conference where the brightest minds and the passionate innovators meet of the pharmaceutical sector. They have gathered and they meet here and they exchange their ideas. I extend my heartfelt appreciation to the organizing committee for their tireless efforts in curating the program of such remarkable depth and relevance. Thank you, Dr. Shara, founder of Academy Innova World, Professor Principal of HR Patel Institute of Pharmaceutical Education and Research, Shetpur, and iSTEM for inviting me to this conference. It is my honor to address you at this pivotal moment because now the pharmaceutical sector is definitely driven by innovation, digital transformation, and global challenges. These global challenges are very much important so that we should take up these challenges to shape our industry and our pharmaceutical sector. I will be presenting my present slide here. Is it visible, ma'am? I have done it in presentation. Has it came now? Okay. Okay. Thank you. Now is it visible? Okay. So here, here we can see that what impact the AI has placed in drug discovery. Here we can see the graph, which visualizes the growth of AI-developed drug candidates. In the year 2016, only 3 drugs have entered in clinical trial. In 2020, 17 drugs, and how it has increased to 67 in 2023. So this is due to the impact of AI in the field of drug discovery. So here we can see that how the exponential growth of AI in the drug discovery pipeline is enhancing our formulation of drug which have been developed by AI. Therefore, the 10 areas of focus of this conferences are definitely very much engaging, and definitely this is the need of the hour. Drug discovery and development, which is being transformed by AI-guided molecular design and data-driven screening, where I have given certain examples which have been generated by AI. One example of case studies has been given. Apart from advances in drug discovery and development, the field of biopharmaceuticals, AI is also having a major impact in the field of biopharmaceuticals, where you can see personalized therapy is also famous. A personalized CRISPR-based editing therapy has been applied to an 7-month-old infant, and the therapy was incorporated in a lipid nanoparticle for gene repair. In the field of precision medicines also, the CRISPR therapeutics is being delivered by nanoparticle, and definitely AI is developing in our formulation of drug, and how it has grown from our previous years to our recent years that we have shown in the graph. Apart from that, the regulatory innovations, they are also catalyzing a faster access particularly for orphan and urgent medications. AI-driven broad-spectrum antiviral and antibiotic delivery is also being generated. In the field of clinical trials also, we can see that AI-driven patient outreach and monitoring expand trial. It is reducing cost and is providing rich data sets. Therefore, apart from these 10 areas, another area is green technology and nanotechnology. Green approaches of formulation and nanotechnology, which is the research area of mine. So innovation is the creation of the new, or the rearranging of the old in a new way. Yes, definitely the statement has been given by Michael Vance. It is not always necessary that always we need to look for a new creation or new innovation. It cannot be always. Rather than that, we have to always search upon the existing literature, and we have to think in a different way, in an out-of-box way, so that whatever are the existing applications, we can work upon it and we can modify it in such an extent that the same formulations or the same excipients or the same polysaccharides or the polymers which were used for a conventional purpose can now be modified for their intelligent applications. So the pharmaceutical industry is undergoing a rapid transformation, which is mainly advanced, which is due to advanced biopolymers, grafted polysaccharides, which are used as a drug carrier, nanoformulation technologies, oral bioavailability enhancement, and innovative drug delivery systems like polysaccharide functionalized nanoparticles. So we know that polysaccharides, already we have a lot of polymer in our pharmaceutical industry. There are numerous applications of polymer which already the conventional formulations are aware of, like gelling agent, binding agent, disintegrating agent, release modifier, and so on. But how can we modify them, or how can we innovate the underutilized polysaccharides? Because we know these natural polysaccharides are present in large amount in our plants, in huge amount in plants. So these natural polymers, they are biocompatible, and it will be helpful for us, for our pharmaceutical sector, if we can extract and isolate those polysaccharides from the underutilized from the plants, and we can modify them so that they can be used to incorporate drug act as a intelligent and smart carrier. In the field of nanotechnology also, nanotechnologies are also suffering from various drawbacks, various side effects. There also, if we can incorporate, we can incorporate polysaccharides for the formulation of nanoparticles, or if we can integrate this polysaccharides with the nanoparticle, definitely this polysaccharide function as nanoparticles, they have the more ability to target a particular area, to target a particular organ, and to improve the physicochemical properties of the formulation. So the topic of today's discussion is natura polyceutics. That is a combination of natural polysaccharides into a pharmaceutical dosage form. It comes under the sector of sustainable pharma and green chemistry, as well as we are going to discuss nanotechnology and drug delivery systems. I think is the full screen visible?
Yes, ma'am.
Am I audible?
Yes.
So this natura polyceutics, they are the emerging science and technologies of nowadays, because they are blending natural polymers with other polymers or with other polysaccharides with a lot of modifications, so that they can be used to design formulation in such a way that they will be able to deliver the drug from the formulation in a smart way, in a stimuli sensitive way, in an intelligent way. Therefore, these polysaccharides, these natural polysaccharides, are shifting of these natural polysaccharides from the bench to market is the need of the hour. Their subsequent commercialization is the need of the hour, as well as these polysaccharides are our today and tomorrow. There are a lot of opportunities of natural polysaccharides as a controlled release. In order to modify the release of the drug from the formulation, they can be used to modify. They can be modified in such a way that they can be prepared stimuli sensitive, either in terms of pH or in terms of temperature. These polysaccharides can be modified in a numerous number of ways. Either they can be blended with another polymer, or they can be grafted, or derivation of we can prepare derivatives of this polymer by carrying out various chemical reactions like carboxymethylation, carboxylation, acetylation, and so on. Depending upon our need, say if we want to improve the solubility, aqueous solubility of the polysaccharide, then definitely we can go for carboxymethylation process. So depending upon the need, where we are going to target the formulation, we can modify the polysaccharides so that we can make them stimuli sensitive. Apart from these release modifiers, they are also having a wide opportunity to act as a biomedical or biological microelectromechanical system. Till now, these carboxymethyl cellulose or other existing polymers, they have been used, but till now, any polysaccharides which have been extracted and isolated from the plant sources, such type of work have not been done in this, or very rare work has been done in this field. So this is a wide opportunity where we can compare with the existing polysaccharides, existing polymers, with the extracted and isolated new polysaccharides. Apart from this, these polysaccharides can also be used to deliver both medicine and imaging agent in a single dose. They can act as a theranostic. This will facilitate real-time monitoring of therapeutic efficacy of the incorporated drugs. Natural polysaccharides, due to their biodegradable nature, their biocompatible nature, they can be used as an excellent agent for this theranostic purpose. Alginate, dextrose, and titosterone work has been done on these polymers, but we need to explore the underutilized ones. Apart from that, we can also go for tissue engineering, formulation of stimuli responsive dosage forms, fabrication of nanoparticles, and fabrication of target delivery system. So we mainly focus, our research area is mainly on these natural polysaccharides, their modification, chemical modifications, and one of the work which we have done is formulation and development of a multifunctional excipient from underutilized polysaccharides, that was polysaccharide extracted from Bacilla alba plant. It was first, the mucilage, that is the polysaccharide from the leaf and stem of the plant, was extracted, isolated, and its chemical modification was done. And this polysaccharide was used to incorporate diclofenac sodium drug in it, and microspheres were prepared out of this. And the safety of, we say that the natural polysaccharides, they are safe, but it is always not so. We have to always carry out these toxicity studies. Even if it is a natural polysaccharide, we have to carry out these toxicity studies. So a subchronic toxicity studies was carried out, and it was found to be safe. And further, these formulations was designed in such a way that this Bacilla alba have already a property, already a gastroprotective property. They have an existing gastroprotective property, and this diclofenac sodium drug was purposely loaded because repeated intake of this drug, this NSAIDs, will cause gastric irritation, GI ulcer. Therefore, these mucilages, these polysaccharides, can be used to load this diclofenac sodium drug in it in such a way that the side effects of this repeated administration of diclofenac sodium can be minimized due to the presence of this Bacilla alba mucilage. The gastroprotective property of the formulation was studied, and further the bioavailability of the drug was checked. In vivo pharmacokinetic studies was done, and their compared bioavailability study was checked. So first of all, whenever we indulge in a research work, already we have a lot of areas, lot of research areas with us. First, we need to identify. First, we need to go through the literature, and then we need to identify the research gap. If we can identify the research gap, then definitely by studying a lot these research papers, this research as well as review papers, we can get an idea that why not this way, why not we try this way, so that we can get a different result which may be positive from the existing one. So we need to first go through the literature study. Then we need to formulate a review article. So a review article was formed on this Bacilla alba mucilage, which can be used as a novel adjuvant in pharmaceutical formulations. And since we have less time, so I have not I have not given the entire details of each and every procedure. Just a little bit thing which I will be saying. That is, first of all, this Bacilla alba mucilage, it was extracted, isolated, and its carboxymethylation was done. In order to improve aqua solubility was improved. Apart from that, various other physicochemical properties of this polysaccharide was also improved. And this diclofenac sodium drug was loaded into it, and the hydrogel beads were formed. It was seen that 80 to 90% of the drug were released in 10 hours time in alkaline medium. So control release of the drug was found. And further, the safety study of the carboxymethylated Bacilla alba mucilages was studied in subchronic oral toxicity study in visceral albino rats, and it was found out all the hematological biochemical parameters were in the range. Histopathological study was also carried out, and it was found out that this mucilages is not having any toxicity, because in our Kolkata, we are taking in this plant, this Bacilla alba plant. It is, in our common language, it is called pui gaj. So we are taking it orally, as in terms of vegetable. So even then we have carried out this toxicity studies, and it was found to be completely safe. Further study was carried out on Mr. albino rats regarding the gastroprotective property of the Bacilla alba mucilage. So here various groups of the rats were taken, where they were administered raw drug, they were administered blank hydrogel beads, and a control group was taken, a group where the rats were administered diclofenac sodium 50 mg per kg body weight were administered. Another group of rats were administered diclofenac sodium and pantoprazole, which is a standard drug, 10 mg per kg body weight. And various studies, tests of gastroprotective property was evaluated in terms of gastric secretory parameters, ulcer index, biochemical tests, histopathology studies, myoperoxidase activity, enzymatic and antioxidant level, their gastric juice, pH, and number of ulcers formed. All these were evaluated, and it was found out that the Bacillus alba mucilages, the hydrogel bead, which was formulated from Bacillus alba mucilage with incorporated diclofenac sodium drug, where the number of ulcerin was minimum, and the number of this gastroprotective property was found to be in an enhanced level regarding all the parameters which have been studied for this test. Finally, the in vivo pharmacokinetic studies was also carried out, and it was found out that the oral bioavailability test, it was comparative bioavailability study was carried out, and it was found out that the formulation was bioavailable. And here we can see the relative bioavailability of diclofenac sodium had been increased from 54 to 96.88% when this drug was incorporated in a Bacillus alba mucilage. So from this study, we have found out that if we can modify the polysaccharides, we can definitely improve the physicochemical property. And definitely this modification is also having an impact on the release modifying ability, the release of the drug from the formulation, as well as it is also having an effect on pH. The toxicity study, which we have already discussed, it has confirmed the safety of this modified mucilage, and therefore we can say that further this type of mucilages from the natural polysaccharides can be utilized, can be modified, and can be used to incorporate such type of NSAIDs drug, so that the side effects of this NSAIDs drug will not be observed, we will not suffer from them. And finally, since it is also improving the bioavailability of diclofenac sodium, so we have got positive results in all aspects of utilizing this Bacillus alba mucilages in incorporating the drug in the form of a hydrogel bead. So that was one aspect of our research work. Another aspect of our research work which we are carrying out, that is nanotechnology, formation of silver nanoparticles. So we know that the chemically synthesized silver nanoparticles, they suffer from lot of side effects. These nanoparticles, repeated this intake of these nanoparticles, either in the form of inhalation and all, they agglomerate, they accumulate in our systemic circulation, and it may lead to toxicity because their elimination is very less. So these silver nanoparticles, they are having a wide range of antimicrobial spectrum. They have a wide antibacterial activity. So if we can incorporate the green synthetic techniques for preparing nanoparticle, then this toxicity of nanoparticles will be eliminated. As well as if we can prepare polysaccharide functionalized nanoparticles. So we have prepared polysaccharide functionalized nanoparticles where we have selected the polysaccharides. They are having some certain extent of bacterial activity, and these polysaccharides, they have a viscosity enhancing property. So if we can incorporate silver nanoparticles in the polysaccharide, so due to the viscosity enhancing property of these polysaccharides, they release the particles slowly into the circulation. So definitely we for any dental applications, because nanoparticle, due to their wide antimicrobial spectrum, they can be used in dental applications. So we can prepare a formulation, a dental oral formulations for this polysaccharide functionalized silver nanoparticle, such that the dose of the silver nanoparticle will also be reduced to certain extent, because the polysaccharides, they are also having a certain definitely very much less than silver nanoparticle, but also they are having some antimicrobial activity. We need to select it in such a way so that the dose of the silver nanoparticle can also be reduced, and thus it can be used in a dental applications, and it can be we are already carrying out this research work in our lab. Apart from that, the biopolymer based nanoparticle, they are having more biocompatibility, they are more biocompatible and biodegradable. Polysaccharide gold nanoparticles, these nanocomplexes, they combine immunomodulatory activity with improved therapeutic outcome in the field of oncology. These polysaccharide-based nanoparticles, they also can be utilized to carry any protein or peptides like insulin for lymphatic absorption and stability in the GI tract. These green synthesized nanoparticles, they offer multi-drug resistant infections and regenerative medicines. So we need, today the pharmaceutical industry stands at a transformative threshold. So this collaboration of academia with another academia, collaboration with other biotechnological industry, with other health departments, with other industrial departments, industries are very much important for this innovative works. So definitely, if we can hold hands and if we can carry out the research work, so we can excel in our sector. So this is my college, Guru Nanak Institute of Pharmaceutical Science and Technology. Myself, Dr. Momita Chaudhary, Assistant Professor of this institute, and this is our working lab where we are carrying out these works. So thank you. I'm extremely grateful and thankful to the organizers of this conference to giving me this opportunity to share these research insights with you. If you have any...
Thank you, ma'am.
Thank you, ma'am. And if we can go for any collaborative work also, so I am always... Yes, sir. Other pharmacy colleges or other pharmacy institutes, if we can hold on hands, because we can see now that how this pharmaceutical sectors definitely a lot of innovations are undergoing here, and we stick with the but if we can share our work with others, if we can join hands with others, if we can collaborate with others, so definitely we can excel.
Sure, sure,
Thank you, ma'am. Thank you, sir.
sure.
Thank you for this insightful session, ma'am. It was very enlightening knowing about the advances we are having in pharmaceuticals nowadays, and it's very vast, and definitely we are looking forward. And now forward for the closer of this session, I would like first to may the Almighty grace be upon us all. And on the behalf of organizing committee, I extend my deepest gratitude to the Almighty for blessing this event. Our sincere thanks to our esteemed dignitaries, Dr. Aswarya Dhar and Engineer Pulap Kumar Dhar, for their visionary leadership. A special thanks to our keynote speakers, Dr. G. Sarvanan and Dr. Momita Chaudhary, for their profoundly insightful sessions that truly embodied the spirit of innovation we celebrate. We are immensely grateful to our collaborators, HR Piper and ISTEM, and to our principal, Dr. Sanjay Kumar Bari, for his unwavering support. My heartfelt appreciation to the coordinators, faculty, participants, and the entire team behind the scenes whose hard work made this conference a resounding success. Thank you all for your valuable contribution. This collective effort marks a significant step towards a transformative future in medicine. Have a wonderful day ahead. As for the schedule, the content presentation will start, and I'm now requesting participants to join accordingly. Thank you all. Jai Hind.
Thank you, sir.
Thank you.
Thank you, madam. Thank you,
Thank you,
sir.
sir.
Thank you, sir.
Thank you, sir. I'm, oh, yes, ma'am. I'm thankful to the Aishwarya Daya ma'am and Punat Dava sir and our respected Dr. S.B. Bari sir for providing this opportunity to collaborate this Innova One and this wonderful conference at a head. Thank you, sir. Thank you, sir. Thank you, Punat Dava sir and Monica ma'am and Sarwan sir. Thank,
Thank you, sir. Thank you, everyone.
thank you.
Thank you. Shall I leave?
Yes, sir. Yes, yes. Thank you, ma'am. Thank you, then.
Okay, have a good day. And thanks everybody for providing me this opportunity to hold this session. Thanks a lot for everyone.